Filtration effectiveness can be ensured by testing the integrity of the sterilized filter before and immediately after use. In the EU GMP Annex 1, the regulatory requirement for sterile medicinal products is required to perform the PUPSIT test. PUPSIT stands for Pre-Use Post-Sterilization Integrity Testing. PUPSIT is a non-destructive integrity test. The test includes the sterilizing filter and assembly, including filter housing, support structure, and connection. PUPSIT ensures that the installed filter is functional during the sterilization and after installation process. There are several types of integrity testing. The three most common are the bubble point test, the pressure hold test, and the diffusion flow test or forward flow. The bubble point test is simple and cost efficient and is supposed to show that the pores of the filter are impermeable for germs and particles of a specific size. A suitable liquid is added to the filter before the beginning of the test. This can be water or an alcohol-water mix, depending on the filter composition. This test measures the pressure required to force a gas bubble through the filter. If the bubble point pressure is too low, it may indicate that the filter has been damaged or is not functioning properly. The second test is the pressure hold test. This test measures the ability of the filter to hold a specific pressure without leaking. The filter is pressurized and held at a specified pressure for a set amount of time. If the pressure drops, it may indicate a leak in the filter. The last one is the diffusion flow test. This test applies a constant pressure of air on the upstream side of a filter saturated with liquid and measures the volume of air over time that diffuses through the filter. Today, the regulators require a stricter separation between operators and product in the form of an access barrier. Isolator technology offers the ability to achieve high sterility levels. The main advantage for isolator-based technology is that it removes the most significant source of contamination from the aseptic environment by eliminating direct interventions by gowned employees. Two systems are on the market: RABS, or Restricted Access Barrier System, and Isolators. But only Isolators are referred to by the US FDA as Advanced Aseptic Technology. Another consequence of the separation of operator and production process is the increased introduction of robot technology in the aseptic environment. RABS stands for Restricted Access Barrier System. It is a physical barrier method of product protection and containment used to control the environment in which sterile products are manufactured. RABS provides a physical barrier between the operator and the product while maintaining an aseptic environment inside the RABS unit. Thus, they have proven to be effective in reducing the risk of contamination. According to the FDA, an isolator is a decontaminated unit supplied with Class 100 or ISO 5 or higher air quality. It provides uncompromised, continuous isolation of its interior from the external environment, that is, surrounding cleanroom air and personnel. There are two types of isolators: open and closed. An open isolator is designed to allow for the continuous or semi-continuous ingress and/or egress of materials during operations through one or more openings. Openings are engineered to exclude the entry of external contamination into the isolator. A closed isolator remains sealed throughout operations. It does not have a direct opening to the external environment. Instead, it uses a rapid transfer port for material transfer. That's all for this session on aseptic processing for sterile drug products. Thank you for attending the training, and I hope you found it useful. See you soon for more Scilife Academy training.